Okay, so welcome. Today is the last class of the single cell RNA sequencing using RStudio. And so today we will go over what left over last time. As first the PCA we will explain a little more. Just a second. And then we will do the cell annotation. And then I will quickly go over the pseudo-time analysis and also the future directions and other considerations about this technology. So let's get some quick review about what we learned yesterday. So still, I hope you show your answers. So if you want to choose A, it's 1, and B, show me finger 2, and C, 3, and D is 4. And I will give you 10 seconds to think about this each question before you give your answer. And so the first question is, which of these metrics is not used for quality control? You have 10 seconds to think about it. Five, 4, 3, 2, 1. Okay. Show me your answer. We have a lot of diverse answers. Okay. So can anyone remind me? Also, we have plenty answers from Zoom meeting. Okay. So what is the feature count? Gene count. Is it all the gene or? Because if it's gene count, all the gene, it should be around 20,000 for each cell. What's feature count?
The genes in one cell, the genes measured in a single cell.
The genes measured that expressed, yeah. So the expressed gene count in single cell. So that's called feature count. And yes, we use that for quality control. And because we can imagine if 0 genes is expressed, like none gene is expressed in a single cell, that's probably an empty droplet, which means originally when we prepare the sample, that droplet doesn't capture any cell, or it can be some technical error there. That's a dropout. So yes, feature count can be used for quality control. And also if a cell expressed too many genes, for example, on average, if the cell expressed 1,000 genes and some cell expressed 5,000 genes, and then that's an outlier, and then we can use that to exclude the doublet, where that potentially the droplet can capture 2 cells or more cells. So that cell is also a outlier that we want to rule out. And the second one, mitochondria RNA content. And do we use that for quality control? Why?
You use it to check to see if there's mitochondrial contamination or to see if there's like evidence of like high cell death.
Yes, that's a great. Yeah, so as we know that one cell is dying, the mitochondria, it will leaking the mRNA to the cytoplasm, and then if we measure too many mitochondrial mRNA in the cell, then it's a sign that's saying this is a dying cell or this is a dead cell. So we also use that as a quality control metric to exclude those dying cells because that can mess up our analysis. And C, so what is RNA count?
The UM.
The UMI count, right? And yes, and that accurately reflect what's originally measured of the mRNA of in that original cell. And yes, we also use that for quality control. And similar as A, if there is zero UMI count, which means it could be an empty droplet and a bad cell. And if it's too much RNA count, it could be a doublet or multiplet. And we also want to exclude those from our analysis. And then the D is not used for quality control. And what is read count? What's the difference between C and D?
To read count, how many pieces of RNA got sequenced?
Is read count is after you sequencing and what's each sequencing you have. Any thoughts? So think, why do we need UMI? UMI is a sequencing, right? It's a 10-digit sequencing that help us to identify the mRNA, the original mRNA. So why do we need that?
To correct for like the PCR amplifications, so if you have like one copy, or like if you see two copies of the same UMI, then you know that it was like from PCR.
Yes, and then those two UMI will be counted at twice in the read count. Yes, so the answer is UMI is introduced so that we can remove the PCR amplification introduced bias where we may see two or more replicates of one mRNA molecule. And then we use UMI, we can identify, okay, these three reads may come from the same mRNA, so we only count it once, and that's UMI count. And if we count them 3 times, and that's a read count, and read count have the PCR amplified signal bias. So that's why we don't use read count. Okay, the next one. You still have 10 seconds to think about it before you give answer. So what is the main assumption made for normalization? Okay, 3, 2, 1. Show me your answer. Mm-hmm. Mm-hmm. Okay, so let's see one by one. So what's the main assumption? So what is normalization first? What is normalization? So after quality control, and we will do the normalization. And how do we do that? So we for each cell, we have the cell library size or cell sequencing depth, and then we will normalize the counts by specific gene over the sum of the gene expressed in that cell, and then times a number. And then that number is kind of what we assumed. We assume that all cells expressing the same UMI or mRNA molecules. So it should be contain the same amount of mRNA molecules, that is the UMI. And still, what's the difference between A and C? So why it's not the same amount of reads? Instead, it's the amount of UMI. What's the difference?
His reads might like count UMIs more than once.
Yes,
So you want like UMIs to be counted once.
yes, because reads can count one molecule twice or more. UMI, because we can backtrack the molecular identifier, so we will only count once. So that's the difference. And here is based on the assumption, we assume they have the same mRNA in the cell. Okay, next question is how many highly variable genes shall we select for downstream analysis? And I think 10 seconds. 5, 4, 3, 2, 1. What's your answer? Okay, so many of you chose B, and that's what we discussed last time. And we also mentioned that it's better to try different values based on your purpose. For example, if you want to include some like immune genes or cancer-related genes, etc., and that if you only chose 2,000 or 5,000 and it doesn't include that genes that of your interest, and then you may change your value, and also you may look at some statistics. So it's better to try different values, and also to test whether your final conclusion is consistent, even you choose different values or very sensitive based on what set you chose. And okay, next. So what is the effect of data scaling? Five, 4, 3, 2, 1. Okay. 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 So what is data scaling? So we know that the normalization is we do some transformation, and for each column, right? Each column is a cell for each cell. And then what is scaling?
It makes the mean 0 and the standard deviation
Yes. So for each
gene.
gene, right? So data scaling is what we do for each gene, for each row. And because some genes may naturally they have much higher abundance, and then some gene, although they may express a few genes, but they're still very important. Just a few mRNA can make a big difference. And so we don't want those high expressed genes overweight or dominant in the downstream analysis. We want equal them for how much they contribute for the downstream analysis. And so because of that, we rescale we rescale all the genes, and they all centered at 0 with the standard deviation 1, and so that it's helpful for our PCA analysis and for the downstream analysis. So it makes all the genes have similar value distribution. Okay. And what is the best description of a metagene? Five, 3, 2, 1. Show me your answer. Okay. Let's see. Here we got some answer as well. Okay. Yeah. So I see plenty of you choose either A or B. So A is one-dimension feature that capture the number of genes. And B is a one-dimension feature that captures the variation in gene expression. And as we discussed last time, the PCA, when we, the metagene is kind of a analogy for the PC, right? For the principal components, either PC1, PC2, or PC3. And because each PC representing a group of genes, and we know that the PC, how do we calculate the PC, is based on the variation. And the PC1 captures the highest variation on that one dimension, and PC2 captures the second highest variation. So here we should choose captures the variation in gene expression. Okay. Mm-hmm. Okay, so does it work? So I'm having some technical issue. Let me try to. Okay, it works. So can the Zoom people hear me? Okay. Okay, so next, great. Okay, and some thing a little more about PCA last time. So some analysis shows that PC1 is strongly correlates with a fraction of zeros. For example, here, if we see that here each dot is a cell, and then here is the fraction of zeros, and then here is the corresponding PC1 score, and we can see some correlation here. And so it's just we can keep this in mind that PC may capture zeros in the data. And then also another thing is that remember that we first selected highly variable genes, and then we do the PCA. And here shows the difference between if we select the highly variable genes versus not selecting. And here is the whole genome. And we can see.
Lou, sorry to interrupt, but we don't see the slides on the Zoom, so it's you're not sharing screen.
Okay, thank you for reminding. Let me try to find out. Share. Let's talk about share. And like, how about now? Can you see? Okay.
Yes,
Okay, great.
thank you.
Okay. So here shows that when if you can see that the PC1 and PC2 the number is a little small, but here in total it explained the 50% variance of the data set using this gene. And if we use a whole genome, it only captures like 5% plus 13%, less than 20% of variance. And so that's another advantage if we select the highly variable genes. And especially PCA is very sensitive to outliers. So yeah. And so we introduced the quality control using the 3 quality control metric. And then we do the normalization based on the assumption that all the cells have the same mRNA counts. And then we selected highly variable genes to exclude the housekeeping genes or all zeros genes, those not of our interest, and only focus on the highly variables one. And then we do the PC. Before we do PC, we need to scale in the data for each gene. We scale them to have the mean of 0. And then we will do the PC. And so that's where we left last time. And next, what we will do is calculate the cell-cell distance and then do the clustering. And so for us to do the clustering, we first need to define the cell-cell distance or similarity. And how we do that is we can use the k-nearest neighbor approach. So that is identify and connect the k-nearest neighbors. And then it will generate a graph. So the k, we can give the parameter k is basically for each cell, how many other cells we call them neighbors. For example, we can use k equal to 10. Then we assume that each cell will have 10 neighbors. And how that's defined? It's defined by Euclidean distance. And we calculate the Euclidean distance, basically the coordinate or the gene expression profile differences between each single cell. And then for each single cell, we can order all the distance between this cell to other cells. And then the closest K cell will be defined as neighbor. And then for those neighbors, we will connect each those cell with the edge. So here shows an illustration. Each dot is a cell, and each dot is a cell, and each edge representing they are considered as neighbor. And we want to use that to define clusters. And however, before that, before we directly using this graph to define the cluster, we want to give each edge some weight, and not all of these edge giving the same weight. And so to give them weight, we're using the GCAR index, which is the neighbor similarity. That is that is for each edge, we will have so each edge is linking two cells. So then we can see the cell 1, the neighbor set, versus cell 2 neighbor set. And we take their intersection and over the union of their neighbors. And this is the number, the number of or the magnitude of the intersection set versus the union set. And then if GCAR index is equal to 0, what does it mean? Yeah. So it means although this two cell has a edge between them, however, these two cells do not have any overlapping neighbors at all. And then it will give you index 0. And how about if the GCAR index is 1? Yeah, it means on the edge, those two cells, they have totally overlapping neighbors. They are exactly in the same neighborhood. And then, so then we have a weighted graph. So for the graph, we for each edge, we also have the weight. And based on that, we can use a Loven algorithm. And that algorithm can optimize the modularity for each cluster for each community. So it maximizes the modularity based on the weight of the edges. And so a quickly understanding of what is modularity. So here, if we consider this as a community and this as a community, and then this will have very low modularity because they are highly connected between these two connected versus within each community. And so this is considered as medium modularity for this community 1, community 2, because it's more connected within each community versus across. And this is a very high modularity where the only connection happens within the community while there's no connections across community. And so that is optimizing the modularity. And there is another parameter. We will see that in the code, and we can carefully check that later. So another parameter will also change how we optimize the modularity. And here is under different parameterization, where we can get different clusters. And that's an example. And now let's get into the code. Okay. Okay. So the code, the last time, we can quickly go over what we where we left last time. So we do the quality control, and for the 3 metric, the mRNA counts. So this is UMI count, right? And then the feature count, and that's the gene expressed for each gene, and also percentage of the mitochondria. And then we use some method to define outliers, for example, using quantile. And then we define outliers. And also we want to exclude those low expressed cells. And also we can see some of the metric. And another, this is with selecting the highly variable genes, is using one line. And then we use this method, and then we can see the red dots is the highly variable gene. And then we also discussed the principal components, and it's representing a set of genes, and the loading for that gene for this PC, it means that how correlated they are. And also we can plot the data within a two-dimension space, and for all the genes on PC1, PC2. And we can also see, like, for the PC1 related genes and what's the cell expression pattern here. And so each column is a cell and each row is a gene. And here only listed 20 genes, and that 20 genes is the highly correlated genes within PC1, etc. And as we can see, the PC1 captures the most variance for the data. As we go more PCs, and then we see that there is almost no variance. They are all like purple, like more similar. And then how many PC we chose, we use the Abel plot and finding the turning point. And we also want to include the genes that we are interested. So here we decided to use 15 for further analysis. And so that's PC. Okay. So that's where we left last time. And so we decided to use PC1 to PC15. And then here we use the K neighbors, right? And we use 30 neighbors here. And here is basically find the neighbors using the k-nearest neighbor algorithm, and then assign it to the object of PBMC. And so this is just one line. And here is we use a Louvain algorithm to define the clusters. And we can specify some parameter. And here is the resolution parameter. And then with that parameter, we can define some clusters. And after we define the clusters, we can have a quick look at the object, the result. And so we previously have this quality control metrics, and now we also have this new defined clusters. So here, for example, this cell, this is the barcode of the cell, and this cell. Okay, so the projector have some problem. Okay. And so this is a cell, and then we can see that it's clustered to cluster 4, and this cell is assigned to cluster 0, and this cell is assigned to cluster 2, and another cell is assigned to cluster 5, etc. And here, this code is doing that if we change the resolution parameter here, if we change that parameter from 0.1 to 1.5, and then we can see different clusters. So a quick explanation of that parameter. So this resolution parameter is basically if it's higher, then with higher resolution, then we will get more clusters. And with lower resolution parameter, we will get fewer clusters. And we showed an example of resolution parameter equal to 0.1, and then we only see 4 clusters, versus if we set the parameter as 1.5, then we can see 10 clusters. And also, if you take even a closer look, that within this higher resolution parameter, that we can see this cluster, the cluster 0, is splitted into three classes, three small clusters. And here the cluster 2 is splitted into two subclusters. And here cluster 1 is splitted into three clusters. And here is by themselves a cluster. And then this one is still one cluster. And what I want to mention is how many clusters you want to define depends on your purpose. For example, if you only care like the resolution at where you can distinguish cancer cell versus B cell versus T cell versus other cells, and this should be sufficient. However, for example, if you care more resolution, for example, you want to distinguish a CD4 T cell versus CD8 T cell, and you want to distinguish, for example, the M1 macrophage versus M2 macrophage versus M0 macrophage, and then you may get into higher resolution. So how many resolution you want to choose, cluster you want to choose depends on your biological question. And also, for example, if this cluster mixing like a cancer cell and epithelial cell, and then that's not sufficient. And also you don't want a cluster include too many different cell types. So that's the resolution parameter, and keep going. And so then here we just tune this parameter and see how it looks like. And here shows the processing of going over that code, of running that algorithm, and after that we can have a look at the data. And here again, that's the first cell, and at resolution 0.5 we have this cluster label, and this means the cell belongs to cluster 4, cell belongs to cluster 0, etc. And can some of you help me to explain what these columns mean? Any thoughts? So what's the title mean here?
The resolution.
The resolution. And what's the value of the resolution? 1.5, right? The resolution parameter equal to 1.5. And what this mean?
The group.
The group for that cell. And so this is the group or the cluster for this cell with the barcode this. And this is for the second cell, etc. Right. Okay. And then here is that we can see how many clusters are there for each resolution. And resolution 0.1, 4 clusters, resolution 0.5, 8 clusters, and 1.5, 10 clusters. And so here and yeah, and then we can now we decided to use this highest resolution. Use the highest resolution for the analysis. And okay, the next thing what we want to do is after reclustering them, we want to visualize them to understand how they are different or similar, or just for like give us some intuition about the data. And what we usually do is using UMAP or t-SNE. And so here so here is a run UMAP, and based on the first 15 dimension, and then we can plot the UMAP. And here Y is dimension 1 to 15. Any thoughts? PCA, right? Yes. So because when we do PCA, yeah, this is the first to 15 PC. And so when we do clustering, if you remember, we focused on the first 15 dimension here. When we do this define the neighbors and then further clustering them, we use only 15 dimensions and after PCA. And the PCA, the purpose of PCA is that we want to handle lower dimension data and for feasibility. And so that's why here we use, so just keep in mind that this dimension should be the same dimension as when you clustering them. So if you cluster, use the first 30 dimension PC, and then here should also be 1 to 30. So it should be consistent between your cluster and then yeah, and then we can see how it looks like for the resolution parameter 1.5. We have 10 clusters, and we have 5 cluster here, 3 cluster here, and 1 cluster here. It might hidden some cluster here, and another visualization we can try is t-SNE. And we also need to focus on the first 15 dimensions, similar as UMAP. And here is how it looks like. And it's more distributed data compared to UMAP. And the UMAP have in this default setting, it has more separation. And that's yeah, and this is a more clear view. And for yeah, and let's go back to the slides. And so yeah, so we do the clustering, and after we cluster, we can see visualize these clusters in different visualization, and using we can visualize them in PC, and then PC you can see that they are not very well separated for this data set. As you can see, this pink is kind of overlapping with others, and some clusters, they kind of overlapping with each other. And the UMAP, it shows better distinction between clusters, and the t-SNE is a different version, and it also shows a better distinction. And keep in mind that PCA is only a coordination rotation. It's
Full green.
the projection has some trouble today. Try to. That's a very random thing. Okay, so can everyone see the slides? Okay. And so for visualization that PC, we can see that it's not very well separation between clusters. For example, the pink cluster is kind of overlapping with all these 2 other clusters, and each cluster, they kind of overlapping with each other quite a amount. And in contrast, the UMAP shows virally, it shows better separation. For example, here is very separated from the rest, and this one is like even they are together. However, you can see they are very distinct on the edge, so they are not overlapping that much in the visualization of UMAP. And on the other side, the t-SNE is spreading out. And like we mentioned, we introduced the PCA. It's just a rotation of the original data space. So this PC, it doesn't change anything, but we just change the angles of the coordinating system of the data. So this PC, it preserves the original distance, and it preserves the original distance or similarities between cells. But keeping in mind, the PC is a projection. So it's from high dimension projects into low dimension. So it's in the high dimension, it might have a very high distance, and after projection, they might overlapping because it's a lower dimension representation. However, it's a linearly transformation. So that's best preserves the distance between cells. On the other side, the UMAP does not, it distorted this distance in the original space. For example, here we can see the cluster is kind of close to this one and this one. At least this cluster should be close to the red-pink one than the yellow-green one. However, in this one, we can see this cluster is more close to this one than this one. So anyway, and for the UMAP, so it's a dimension reduction technique. And so it use predominantly, it's mainly used for visualization. And it the principle of this method is capture the manifold or the topology, shape, structure, connectivity of the data organization. Here shows an example of the UMAP versus the PCA. And if you think of this as a high dimension gene expression space, and this is how each dot is a single cell, and it presents how the single cell distributed in this space. And then the UMAP can find an axis, a low dimension representation. It will find this low dimension. So it's the red one. So the red one is a curve, right? This red curve will be one axis in the UMAP, and so that it's best capture the topology and most also the data variance. However, if you do PCA, as we know that PCA is only linear transformation, so you will find another axis, which is the blue line. And then that blue line keeps the distance, but also it does not capture as much as variance as in the UMAP. And so just 2 points to highlight is first is very successful on visualizing the data for UMAP and t-SNE. However, we only use it for visualization and never use it for clustering. We only use PC for clustering because that best preserves the original distance between cells. Another thing is that we can also tune the parameters of the UMAP. Here shows some, for example, the neighbors you can define, and also you can give the minimum distance for UMAP. And then for different parameter, you can see totally different patterns for UMAP. And this is under one parameter, and you see very distinct cluster. And this is under another clustering, and then you kind of see although they are still separated, but they more close to each other under this new parameter setting. So this is another point of why we use UMAP only for visualization, but not for clustering, because this can create something that's not true. It's just for our understanding how the data distributed. So next thing that what we will do, the projection is problematic again. Okay, and next, let's go back to, oh, I think it's time. Okay, let's take a 10 minutes break, and then we will come back at 10:05. And during your break, please fill in this survey, and it's important for our improvement. Okay. Where are we? Okay. Okay. Let's get back to the class. And check something. Okay. So after we have these classes, and then we're projection having problem again. Okay. And then what we want to do is identify the marker gene, and because because this helps us to identify the cell type, and with these marker genes. How do we do that? So this is basically answering the question, which genes give the identity to each cluster, and how do we define marker genes? So marker genes are the genes that are differentially expressed in one cell population, one cluster, compared to all the rest. So that's specific to this cell type, and that's marker genes. And and CRAT performs the differential gene expression analysis using the Wilcox rank sum test. So it keep having this projection problem, and let me try to fix it. Okay.
So we can't see the slides in the Zoom. I'm sorry. We can.
Can you see?
Yes. My mistake.
Can the people on Zoom see the slides?
Yes. Yes, we can.
Okay. The projector keep having problems, and that's why it sometimes goes off the share screen. So just let me know if you have any trouble to see the slides. And so yeah, marker gene is that's specific to a cluster. For example, here shows an example. This gene only expressed within this cluster, and the CD14 only expressed in the cluster here. And so first to do this, we first need to identify the differential expressed genes. And CRAT already do that for us using the Wilcox rank sum test or the ROC test. And so I also want to highlight that the marker genes is how do we define like significant different expressed. Here shows two example that the blue is one group and red is another group. And if their mean are very different with their spread not overlapping, and then we can claim that this is a significantly differentially expressed gene. And so how do we decide whether it's significant or not? And we use those statistical tests such as Wilcox rank sum test and other tests. And here is another example. Although sometimes we may see the gene expression average can be different, such as the bar here. However, their spread, the variance of the data is high compared to the mean difference, which means that these two groups are they have a lot overlapping. And then we won't be able to really distinguishing these two these two groups just using this gene. So then we won't consider as a differential expressor genes. So that's what we are doing for the test. And let's go back to the code. And here is the visualization we just discussed. And here is using, we can visualize different resolution results, different resolution clusters results using UMAP. And here is a low resolution cluster and higher resolution clusters. And this is what we are processing now, the resolution 1.5 with 10 clusters, and we can see more higher resolution. And now what we will do is finding differentially expressed features or genes. And so first, here using this low resolution as an example to identify the gene markers and the cell type markers, and we just use one line, and here it shows that for this CRAT object, we gave them the first cluster and the cluster 4 as the first we want to compare, and then cluster 5 as the second cluster we want to compare. And then we only focus on the genes that at least expressed 25% for either cluster 2 or, sorry, cluster 5 or cluster 4. And then after we run this line, and then we can assign it to the marker data frame. And here we then can have a look at this data. And here shows the marker genes. So this is different gene showing here. And then this is the p-value of the fine marker. They're using some tests. By default, they use the Wilcox rank sum test. And so this is the p-value for that test. And then this is the average log2 fold change, which means the fold change of the second given cluster versus the first given cluster. And sorry, the first over the second, the first over the second. So if it's negative, because this is taking the fold change, taking log2. So if it's negative, what does it mean? What does it mean?
It's higher in the second than PCT2.
Yes. So if it's a negative value, it means the log2 fold change is less than 0. It means the fold change is less than 1. And the fold change is equivalent to the first given cluster over the second given cluster. So it means that the first is less than the second. And then we can also see that in this metric, the percentage of this gene for the first given cluster is only 9%, and the percentage for this gene for the second given cluster is 85%. So this is much higher expressed within this cluster for this gene. Great. And this is a adjusted p-value, and it's used when we do not pairwise test, but multiple test. So then we need to adjust the p-value. And similarly here, can any of you explain to me what does this line mean? What does this row mean?
This given gene is higher expressed in PCT1, PCT2.
Mm-hmm. With the fold.
Change being positive.
Yeah. Mm-hmm. Yes. Uh-huh. So this lines means that the CD3D, this gene, is significantly higher in the first cluster compared to the second cluster. And how much high is given by this number, like the fold differences of log 2 of the fold differences 3.96. Any question?
I have a question.
Mhm.
So is there an actual p-value in the output or test statistic so we could calculate the highly significant p-values?
Yeah, here is the p-value is 0. It means it's below the machine the smallest number that machine can process.
But for example, for publication, you can't say p-value is 0. So is there a way to calculate the actual p-value? Is there a test statistic in the output, or is there a way to get an exact p-value?
Yeah, this is the exact p-value, and these genes are, because this data set are well designed for this tutorial, so that's why this is so perfect. So in the other data sets, this won't be 0, or for most of the case, unless it's a perfect marker. So a lot of markers, you will see this value not 0.
No, but what I mean that can we get the actual p-value somewhere like 10 to the negative 300 or whatever it is?
Yeah, it will be given in this data.
I see. Okay, thank you.
Yes.
And when we will export this data, so we will get original number in the Excel or CSV file.
Yes, yes. You can explore this data. Yes.
Sorry, could you calculate again how you, so is this significance and the log 2 fold change derived from the same value, but it's just different tests? Or what determines log 2 fold change and what determines significance? Is it both just the deviation from the mean expression?
Yeah, that's a statistic test question. The p-value is usually comes from the p-value comes from the statistic test where you have a null hypothesis. Usually we assume that these are exactly the same. They are not differentially expressed. And then what's the probability we observe the current data? And then that probability considered as p-value. And so it's basically under some null hypothesis. And then this one is, I'm not sure how this calculated. If we using statistic to calculate that, we can calculate the effect size. And one way is doing that is like using like bootstrapping, or if if you already know the underlying true distribution, you can also use the true distribution. Usually that we don't know. And then you basically subsampling from the data and then calculate the confidence interval. And then the median you already can report it as the, so yeah.
I have a question too, real quick. Sorry.
Yeah.
This is basically showing a cluster of the same samples, right? And what I mean by that is let's just say you have one tissue that you're looking at and you're comparing endothelial cells to, CD4 cells or however. Can you also apply the same method if you have two independent samples, like if you have treated versus non-treated? And if you were able to identify clusters, can you then kind of mix and match?
Yeah, that's a great question. And we need to pre-processing the data first before we compare them. Because we know that samples could have like batch effects. Yeah, different yes. And yeah, so we need to integrate the data first. And that's gonna be shortly covered later, how we do that. And then after we integrating them, we can use this one, yes.
Okay, perfect. Thank you.
Mhm. And and then what else we can do is that we can also not only comparing like one cluster versus another cluster with the differentially expressed genes. We can also compare one cluster versus another set of clusters or all the rest of the cluster. Here we only specify 3 other clusters, and then we compare, okay, what's differentially expressed between these two group. And then also we at least need this gene is at least expressed 25% within either this one or this one. And then it listed genes here. And this is still the p-value. And I believe that if you check into this object or this data frame, and then if you can display that to some digits, and then you may see non-zero values. Here, I think it's because the machine is not detectable to small values, so it's 0. And then here we listed the genes, and we can see the average log2 fold change is positive, all positive. And for example, here is for the log2 is 5. So what's the real fold change difference? If not taking log2.
You just take the ratio of PCT1 and PCT2.
That's a great approach.
Is that wrong?
I mean, that's the intuitive
I guess
way to calculate.
Are these real values or are they like?
These are real values. These are real values. Yeah.
There's two component values of the functional expression. PCT. PCT means there's two component value or original expression value of the normalized value.
If this is percentage of a cluster, the first given cluster or the first set of a cluster, and the percentage of the second for the corresponding gene, expressing that gene.
After normalization, the percentage.
Yes.
During the normalization also, the number will be changed. The original expression number will be changed.
Yes.
During the normalization.
Yes. Yes. And yes. And so this one is just a 2 to 5.6, right? 5.6, and that's the fold, the original fold change before taking log2 transformation. And then we can see that the percentage this gene is highly expressed in the cluster 5, the first given cluster, 96% of the cell expressing that gene. And then this is the second cluster is actually a set. It's 3 cluster together. It expressed only 8%. And similarly for the rest. And so that's how we identify the that. And next is that then we can use so here we just compare by pairwise, right? And what we can also do is just for each group we can identify the markers, and then this is just one line, and then we need to give the minimal expression percentage, and so this is the one, and then we can assign it to this data, and yeah, and here is basically we can select a subset of all the markers, and so we want them grouped by clusters, and also for each cluster we want to display 2 markers, and they ordered by the log2 fold by this value. And then we can display this how it looks like. And first, this is a p-value, and it's because it's the top 2 marker for each cluster, so it's still very low p-value, 0. And then here is the average log2 fold change comparing this cluster. Where is this cluster 0 versus all the rest. And here is similarly also this CD40 LG is also specific to cluster 0 compared to all the rest. And here is a percentage expressed in cluster 0, and here is percentage expressed in the rest of the clusters, in the other clusters. Similarly, we can see here. And then for cluster 1, here we also see like CCR7 is a marker genes for cluster 1, and with the log2 fold change 2.5, and expressed in this cluster 1 with 50%, and for all the rest cluster is expressed 10%, etc. And so that's how this looks like. And another, so by default, it will use the Wilcox rank sum test, and we can also use some other test. For example, the ROC test. If you are familiar with statistics learning or machine learning, so this is usually quantify how well a binary classifier is. And basically you train the classifier, and you can plot the false positive rate versus the true positive rate. And then that's the curve. And then the area under the curve can give you the metric of how well that classifier is, how good that classifier. So yeah, and here they're using this as a test to identify whether we can use the marker to tell us whether it's specific cell type, and so you can also try all different statistic test, and similarly we can display using this method what genes we can identify, gene markers that we can identify, and here give us totally different genes. For example, here is different than the another test. So this is also something that you need to consider for based on your data, and also which one gives more reliable results based on your biological knowledge. And after that, we can do the feature plot where, so here is after we identify the marker genes, and then we can visualize the data to have a look And here is how we look at them. So let's focus on the RRC test identified marker genes, and then we display these genes and for this object, and then using this function to plot. Here is a marker genes for cluster 0, and let's go back to see where is cluster 0. So we are focusing this one. So cluster 0 is here. Cluster 2 is here, and oh, sorry, cluster 0, 1, 2, 3. Okay, and so here shows a cluster 0 and cluster 0. So this is a good marker for cluster 0. And this one, it has some, but it seems also have some in the other cluster, similar for this one. And so, and this is seems for cluster 2, and this is for cluster 2. So this is for cluster 1. And so this is for this cluster. Okay. And etc. So it identified some genes, and yeah, so this is some not good genes that identified by this test. Yeah, it doesn't have a cluster. Okay. Yeah, there is some issue here. Since we don't see anything. Do we see anything? Yeah. And here is so how do we read this map? This is a U map, and you can display all the clusters here, and then the color means the corresponding gene. The higher means higher expressed or higher density of the cells. And then so here we can see the gene expression pattern among the cell population. Another visualization, what we can do is using dot plot, and then we still plot this set of genes. So these x-axis shows the genes that we identified as marker gene, and then the y-axis representing the cluster identity, and then we can see and color means the average expression, which means for example here if we have cluster 0 100 cells, and then on average this 100 cells it only express 1, and then that's so then the average expression is showing by the color, and then for example 50% of this cluster expressing this gene, and that's identified by the circle size, and so this is the dot plot, and if we identify really good markers, then what pattern would we expect to see? If these are really good markers, what pattern would we see? Any thoughts? Diagonal.
Yeah.
So if this is a really good cell marker, then we would expect it only have values on the diagonal. And on the diagonal, we will we will see high color, a high expression, and which means the color, and also the large circle, which means higher percentage of the cell expressing that gene. So and then we would see a kind of a line pattern. And so here we can see that this is highly expressed in 0. However, it's also expressed in 1, 3, 4. So this gene is not that specific to cluster 0. And so we need, and then you can, what you can do is you can try different tests and see whether it may select a different set of gene. And also some tests you may give some different threshold or values and see. And here for, so this one should be the cluster 1 marker genes. And we also see that's also not very specific. And seems like the cluster 1 and cluster 0, they are very similar in terms of this marker gene expression. And however, like here, this one, the 5. So these 2 genes are the cell marker genes that we identified for cluster 5. And then we can see this seems only expressed in the cluster 5. And not only highly expressed, but with a high expression percentage ratio expressing. So that's a good marker. This is a good marker, etc. So here is the good marker. However, this one is a marker that yeah, you need to carefully to check. And so that's one another plot. How we can visualize this is using heat map, and then it's just this function, and then you run it, and then you will see this. And here still cluster 0 to cluster 7, and each column is a cell, and each row is the gene that we identified as marker genes. And so these two we using test saying that, okay, this is a marker gene for 0, for cluster 0, and then we can see it's fine. And then this one is for 1, and that's terrible. And so this 2 gene, oh, okay. So this 2 gene, this 2 gene is supposed to be cluster 2, right? And that's a pretty good cell marker, because the color means expression in that cell. And then we can see that cluster 2 cells, it highly express these 2 gene markers. And for all other cell types, or for all other cluster of cells, it doesn't express these 2 markers at all. And so if this is a good set of gene markers, what would we expect to see? Yeah. So this pattern of is only yellow color in that cluster, and all purple color for the rest cluster, for the other clusters. And also because if you order them by cluster, like the cluster 1, cluster 0 marker gene, cluster 1 marker gene, cluster 2, etc. If you order, then you would also expect seeing a diagonal pattern. That's only on the corresponding diagonal is the yellow, and all the rest are the purple. And another way to visualize these marker genes is using violin plot. And here shows these marker genes. And this is marker genes for cluster 1. And also we already see that this is not a specific marker, because it also expressed in cluster 1, both 0 and 1. And others, it is less expressed in this gene. And so if a gene, if the a marker gene is well defined or specific, what would we expect? What pattern would we see? Or can you tell me which one is the best marker?
We want the layer.
What's the name for the
For
gene?
example, the 7 is the L6, 6,
This one?
so consider
Uh-huh.
6.
6.
6 is the graph lower level,
This one?
left to second.
This one? Okay. Uh-huh. Yeah. This one can be considered as a good marker, and it's highly expressed in this cluster, in 6 cluster. And similar, this one is also can be considered, and because it's all zeros for other, and only distribution in cluster 6. Yeah, and similar, this one and this one and this one is also good, right? And this is terrible, and this is yeah, you can tell some information, but yeah, it's not perfect. And sometimes, here the data we don't have that sort of data, but you may also use 2 or more markers to identify. For example, here if this one it shows highly expressed in cluster 0 and cluster 1, and then imagine another gene it highly expressed in 1 and 2, but low expression for all others. So this is highly expressed. Imagine highly expressed in cluster 1 and 2, and this is cluster 0 and 1. And then use these 2 genes. Which cluster can you identify?
Wait, sorry, can you repeat the question?
The question is that I can draw it. Let me check the Zoom people whether. So you can imagine that this is the violin plot, right? And cluster 1 and cluster 2 and cluster 3. And then you have a marker, let's say gene A, and that's highly expressed. And also highly expressed. But this one is 0. And etc. Cluster 4 is also 0. That's gene A. And then gene B. Gene B is that this pattern where this one is highly expressed. And then this one is highly expressed. And then all others are 0. C1, and C2, and C3, C4. And then this is gene B, and this is gene A. And then using gene A and gene B, which cluster will you be able to identify?
Like as different. So you can use
C1.
C1 or C3.
C1 or C3?
If you look at both of them.
Yeah. So use both of them. You can any other answer?
You can separate based on C1, C2 minus C2, C3. Is there any chance? We can we separate C1, C2 minus C2, C3?
Yeah. So actually, it can distinguish these 3 if you analyze them properly. So first, if a gene express both A and B, if a cell expressing both A and B, what cell type would you classify it as?
C2.
C2, right? C2, C2, because this both A and B is highly expressed in C2. Okay, and if a cell is only expressed in B, and what cluster would you expect it belong to?
C3.
C3, right? C3. And now, and because if a cell is highly expressed in B, then it's either C2 or C3, and it doesn't express A, so it cannot be C. It's probably not C2, right? Because if it's C2, then it should highly express A. So then it's probably just a C3. And etc. So actually, and if it's A minus B minus, what's that?
C4.
C4, right? C4. Right, because it doesn't express anything. So actually, two markers can help you to distinguish. In perfect situation, it will be able to help you distinguish all four clusters. So what I want to emphasize is that although this maybe not like look like this is not very clean, however, it still provides you information, and you can combine with your data, like the case here, and then you maybe still be able to combine them to identify some specific cell types. Yeah, that's yeah, great. And then we can assign the cell type identities to the clusters. And what we usually do is that, so based on the markers that we identified, and then we can compare with either the library, the cell marker library, or your own knowledge for example, some subtype or some new cell type that's not well documented in the library, but you know that, okay, this should be specific to this immune cell type. And then you can use your own knowledge and library, and also use some like AI chat box like ChatGPT to help you identify the cell markers. And to assign each cluster to a cell type. And so here is just manually we assign each cluster to a cell type. And for example, here by checking the library, we realize that this is a CD4 T cell and etc. And this is a B cell, and this is a dendritic cell, nature killer cell. Etc. And then yeah, so then we can label them as well. So based on the cell marker, once we decide the cell types, and then we can assign these labels and to the object, and then we can label them in the UMAP. And here shows that. And then we can have a further look at the data. It's still the, yeah. I think that's and yeah. In this manual, the last part is just you save this for because all of these analysis result is saved in PBMC. So you just need to save this object, and next time when you load this one, when you read this object, and then you don't need to repeat this analysis again. You can just directly use the result. And so if we go back to the slides. Oh yeah, this is the ROC curve. And yeah, this is some how do you identify the cell types? You can list some tools like cell marker 2.0 and marker gene annotation and ChatGPT, etc. And so
The triangle three, or
sorry,
the second one.
the second one. Yeah, it's a tool. That's
So what is the input data?
you oh, sorry,
What is the input data?
the genes.
Excel, CSV file.
Yeah. So for all of this is that you already know the so you identified some marker genes, right, for each cluster, and then you want to see what cell type for each cluster, and then you input the gene name, and then they will tell you, okay, this marker seems like T cell, not seems, this should be T cell, this should be B cell, this should be macrophage, etc. Okay. And so let's do some quick review and still think 10 seconds and then show me your answer with 1 is the A and B is 2 and C 3 and D 4. So first question is, what algorithm is used to find neighbors of a cell in the PC space? Five, 4, 3, 2, 1. What's your answer? Okay, yes. So for us to find neighbor, that we use the k-nearest neighbor algorithm. And what do we use Loven algorithm for? Clustering, right? We use so this is the algorithm we use to identify the clusters. And how about Jaccard index? What do we use it for?
Measures the overlap genes.
The overlapping neighbors. Yeah. So it's basically tell us it's basically give the weights. So after KN, we will have a graph, see whether they are neighbor or not, right? Two cell are neighbor or not. And then, however, not every edge we want to use the same way. Some edge we give them higher weight. And then how we give the weight is using Jaccard index. You remember we using the intersection of the neighbors over the union of the neighbors, right? And how about UMAP? What do we use UMAP for? For visualization, yeah, reduced dimension visualization, right? Can we use it for cluster? No. Why?
Original dimension will be lost.
Yeah. So the original distance matrix will be distorted. So it doesn't preserve the real similarity between cells. Okay, and great. Next question is what is a marker gene? Five, 4, 3, 2, 1. What's your answer? I see. So some of you chose B, some of you chose D. and marker gene. So let's first look at B. A gene differentially expressed in multiple clusters. And D is a gene whose expression helps identify clusters. Yes. So yeah, usually when we define marker genes, we define that compare this cluster versus all other clusters, and what's the differentially expressed gene, and that's how we define the marker gene. So it should be D, and this is a little like too vague because it could be like if I compare like cluster 1 and 2 versus 4 and 5, and then usually we don't call that marker gene unless we define cluster 1 and 2 as one cluster. And but still keep in mind that there are genes, combination of genes we can help us to identify the cluster. Yes, yes. Okay. And yeah, let's take a break and we will start from 11:05. And yeah, we will quickly go over the pseudo time and then some other directions in the field. And yeah, so let's take a break. And if you haven't signed, please sign your attendance. Okay. Mm. Let's continue. Next, what we will go over quickly is the pseudo-time analysis. And how many of you have seen this picture? Mm-hmm. And what does this mean?
the line landscape, which is like at a certain point in time, a cell will differentiate or a cell will become differentiated in a specific time cell. You can't like differentiate in any other cell.
Yeah, great. So this is a Worthington landscape, and it's kind of visualize how we can think of this as energy landscape, and then it's and each cell is going only on specific path, and till specific point it will differentiate into and goes into different cell fates, and then it may further differentiate, etc. So this is just building an intuitive understanding of if we using some physics understanding, then how this is, how cell is differentiated, how that's controlled, and this is a landscape to help us to build the intuition. And so the pseudo time is kind of like similar. It's basically reconstruct the path of cell differentiation from the single cell sequencing data. And so there are some assumptions about the pseudo-time analysis. First is that the development of cells can be modeled using a smooth, continuous surface. And yeah, that's the algorithm, the underlying algorithm. They have to based on that assumption to work. And all cells are undergoing transition. It means that this single cell, it can goes from, for example, the hematopoietic stem cell, and it can goes toward to the aerial one cell lineage, and also another like lymphoid cell lineage. And all the cell we observed is at one state along this path. That's so it's basically all cells is undergoing transition. And so, and also another thing is they have similarly trajectory speed. It means that how long does it take from the point to here should be equivalent to how long does it take from here to like here-ish. So the speed, it should be similar. And so here shows some paper that using pseudo-time analysis. And this is the pseudo-time algorithm that we will focus on and use. And here shows the data of the fertilized egg, the development of the development of the egg, and then using this analysis on that data set and what result it gave us. So from the 1 cell to 2 cell, 4 cell, into 16 cell stage, etc. And using this algorithm, the reconstructed like pseudo time developmental time, and yeah, it roughly aligns with the biological development time. And So here is some yeah, here is some also further is that yeah, so we need keep in mind that the pseudo time is an abstract unit of progress. It doesn't equivalent to the physical time, and sometimes it may align. For example, this one, it may align with the developmental time, but most of the time it is not equivalent. And so it's defined as the distance between a cell and the starting point along the shortest path trajectory. And so let's quickly, I shared some code on the Google Drive, and yeah, yeah, you need to install some library and also use a specific data that they provided you to run that code. And here, just to quickly, I just showed some tutorial that's let's go to the results part directly. So the beginning part is mainly what we've done, like identify gene markers, etc. And so to do the pseudo time, so here this one, the CDS is the object, and here is that we're showing the clusters and using what we just learned, like the Lewin algorithm to clustering them and UMAP to visualize them. And here is the cluster numbers, and we visualize them. And then here is that you use this function to calculate the pseudo time trajectory, and for this object. And so this is equivalent to our PBMC. It's basically the serialized object. And then you, after you learn that, you assign it to this object again. And so once you've done that, and you can plot the nodes. And here it shows that we want to have a look at the branching point and the roots. And here, this circle is the nodes that it identified. And so it's either roots or the branching point. And then another thing they can do is that we can order that and also plot them. And this and plot them in the like show the pseudo time. So here we can see it's starting from here, pseudo time 0, and then it gradually develop along the pseudo time as the time color goes up. So it gives us a trajectory. And etc. And so here is using some this is another algorithm that we can use to produce the pseudo time. So then you just use this function, and you can get that, and you can get that data. And after you get, you can plot it, and here shows different clusters. And this shows for each cluster, we have different cells, and each cell, it has a pseudo time assigned it to it. And then we can plot for each cluster what's their pseudo time value distribution. And here is how it looks like. And this is ordered by the cluster. We can also order it by the pseudo time. So if we ordered by the pseudo time, and then we can see that it's a smooth transition, and it gradually goes up. And also, yeah, and then, yeah. yeah, there is some other functions you can also explore that if you are interested. For example, how each gene distribution change based on the pseudo time and the clustering. And here is ordered by the gene expression pattern from low to high. And then you can also plot that gene expression dynamics along the pseudo time. For example, this gene is first highly expressed, and then as time pseudo time goes, it goes down first, and then it goes up, etc. And this is some thing that you can explore. And this is another tutorial, and here something that you can also show. Here is a nice figure of showing this pseudo time along different lineage, and here is a erythroid lineage, and this is a lymphoid lineage, and this is like pseudo time 0, and it's gradually differentiates into the most differentiated stage, and similarly this one. So you can also plot that. And so that's for the pseudo time. And the others is there are also other directions. For example, we know that we have batch effects in the single cell data, and so even like they biological replicate, and if we just do the experiment separately, then we may get totally different like UMAP or the visualization of the data basically. And what we can do is that we can do the data integration, and that's already also written in the CRAT, in the CCA function. And briefly explaining the principle is that they find the gene expression correlation, and then they can, based on the gene expression correlation metrics, and then they can transform one data to another, more similar to another data. And in the transformed one, they can identify the anchor cells, which they are the most similar in the gene expression. And then using those anchor cells, then they can mapping all the other cells distribution. And then this will give us a pretty good data integration result. And so here we also can have a look at the data integration. Here is a function that we can use. It's called integrated layers. So this is the data for different experiments. Some of you may ask that what if we do different experiments? And so here is control group, and this is stimulated group. And as you can see, if you directed UMAP plotting them, they are like not overlapping at all. And then, so we need first to do the data integration. And how you do that is you use this function, and the method you can use this one, the CCA integration. And the reduction is still PCA. And then you assign it to the object. Yeah, here is find the neighbors, and then find the clusters. Yes, this is just joining the data. This is basically the data integration codes, and you can see after you integrating them, they are overlapping pretty well. So it potentially remove the batch effects based on the algorithm. And so this is control, this is stimulated, and now you can compare them after data integration. And using like what we just did, identify the gene markers, and for or identify like what different expressed in the macrophage for this condition versus macrophage within this condition. And you can do that, the differential expressed genes. And some other analysis is that you can also do the gene ontology. And basically, after you identify differentially expressed genes, and it can be a full list of these different expressed genes. And then you may want to know, okay, what this gene is doing. And then you do this one, and then it tell you, okay, what you specify that gene is the function is sensory perception, skin development, sensory perceptual smell, etc. It's just tell you the functions of the genes that you identified. And also you using the single cell RNA sequencing, you can do the RNA velocity. So this one is basically telling the derivative of the RNA. It's basically you measuring the RNA now, and then you know the tendency, the next time point, how it's gonna develop. So this is RNA velocity. And the principle is that they using the spliced and unspliced and their ratio, and then they can define the velocity, the rate. And this is one of the results that I love the most. And so here you can see, this is a very beautiful pattern. Here is a spliced RNA-seq, and each row is a gene, and this is unspliced. And then you can see this is time 0, time 3 hour, 6 hour, etc. And then you can see the unspliced gene expression pattern precisely, accurately predicted the spliced pattern in the 3 hours. So it aligns with the biological process that the mRNA first become transcribed and then spliced, and then etc. And so you can see that actually in the data. So that's very cool. And then they can use that ratio to define the velocity, the rate of the change for different genes. And other thing that you can also do is the cell-cell communication analysis. So this, you input the single cell RNA sequencing data, and then they will, based on the library of the ligand and receptor pair, and then they basically, the assumption is that, okay, the ligand mRNA, it can infer the real ligand secreted by that cell, and then the receptor mRNA can also infer the real receptor abundance on that cell. So if one cell expressing the ligand mRNA, the ligand A, and then another cell expressing the ligand A receptor mRNA, then they infer that this cell have some communication between them. So that's the principle. And then why this is useful, because we know that all the cells, they don't function by themselves. We are not a single cell organism. We are like full of different cells, so they coordinate with each other, and so they need to communicate. And for example, the macrophage, it senses the immune threats, and then it's like a leader, and then it detects bacteria, virus, or damaged tissue, and then it tells other cells, oh, here is an immune threat, come to join me, let's defeat this together. And so so yeah, that's what cell communication can help us to understand, to really see how cell they coordinate with each other. And can the Zoom persons, do you see the slides? Okay. 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 And yeah, that's one. Another cool paper is this cell atlas that just come out last year, and basically they collected 20 million cells profile mRNA sequencing, and then you can input your mRNA sequencing results, and then they will tell you what's the most similarly cell it is belongs to, and it's a macrophage, it's a T cell, and if it's macrophage, what's it's like cancer related macrophage or yeah, the projection has problem again, and etc. And so. Yeah, and then you can you can then input, and then it will give you the information about this cell. Yeah, and other considerations, if you're doing a wet lab or you're designing, whether you, and it's the cost, like the sequencing, it may cost differently. It's yeah, and also the lab preparation, it also costs differently. So those one you need to consider. And also the lab preparation, you need to read carefully at the center, like what they require, because each one may be different, and also another is the current trends in the RNA sequencing analysis research. And first is very hot now of the single cell spatial transcriptomics, and yeah, and one thing of in the beginning, we mentioned in the beginning of the workshop, we mentioned that the advantage of single cell RNA sequencing versus the bulk is that it can help us to identify different cell types, and it can help us to identify cellular heterogeneity. And the limitation, what's the limitation? Expensive, and besides that? Spatial, it doesn't have a spatial, yes. And if you compare single cell versus bulk, what's the limitation of the single cell? If you comparing the bulk RNA sequencing versus single cell RNA sequencing, what's the limitation of the single cell RNA sequencing?
To capture less RNA.
Yes, yes. Because bulk can capture 90% or more RNA, and single cell, it only captures at most 30, the range from 5% to 30%. So it's much more noisy. And so similarly for spatial, yes, single cell doesn't have spatial information, and spatial have spatial information. However, spatial transcriptomics covers even less. It's I would say like less than 1% or 0.3%, something, some number like that.
So cells get analyzed?
Sorry?
1% or to 3% of what?
1%, less than 1% of mRNA captured by the spatial transcriptomics.
Total of the tissue, or what you are comparing, 1%?
How much mRNA is captured in that technology? Like the cell, it have 100% of mRNA, but not everyone is measured, is captured, is observed. And in that technique, only less than 1%.
One cell. You
mm.
are mentioning about one cell. Oh, per cell, how much mRNA?
One
You
cell.
are mentioning percentage as the per cell mRNA percentage.
Yeah, on average, per cell.
Okay.
Yeah, per cell. Yeah. And then, so, yeah, so you can imagine that when they do the spatial, now they do 2D, right? And they need cut like 10 micron slides. And then a lot of cell, it doesn't always align like a layer perfectly. You always cut like part of the cell. And that's already like contribute to lower capture. And also the technique doesn't like even you capture the full cell, it doesn't guarantee you capture everything because the spatial you need to preserve that information. Yeah. And so, but it still provides us very interesting informations. And what currently they do is they using single cell to infer or to like they matching the spatial transcriptomic and using a single cell label transfer or infer some information of the spatial.
Is that how they, so this Allen Brain Atlas, I'm not sure if you have seen it, they have like pieces of brain tissue
Mhm,
and they
mhm.
have, did they do that where they took like the single cell and they inferred onto the spatial?
I'm not sure which the research you refer to, but I know that plenty of them doing that. That the when they do the spatial, because it's really sparse data, it's less than 1%, and then to really get some information, they need to like link to the single cell and to either label transfer or doing something more analysis. And another tendency is that the single cell RNA-seq and also single cell ATAC. Do you know what is ATAC?
It's a type of like chromatin. I can't remember what it stands for, but it looks at like chromatin remodeling.
Yes, yes. So it's the chromatin accessibility. So if the chromatin is opening and it's not binding the histones and it's ready for transcription, and then that part can be captured, and that's ATAC. And as we know that the ATAC is kind of the starting point of transcription, and the RNA-seq is the readout or the output of the transcription. And so they can mirror, there are technologies that mirror them together for the same cell. And also the long read single cell. So what we have been discussed is mainly the, if you remember, the when we are discussing the length of the mRNA, it's only 98 base for the sequencing. And so 16 base is barcode, 10 base is for the UMI, and then for the mRNA part is only 90, yeah, it's less than 100 base. And so they have long read, so they can read the full mRNA length, like it can vary to like thousands base. And yeah, that's also a direction, and also cell-specific genetic regulation, and that's use the algorithms. And yes, I think that's all. And the homework is in the Google Drive already, and I will upload today's slides. And also, I will send out a quiz for those people who want credits, and for those who don't need credits, and you feel free to do that or not. It depends on you. And then the deadline will be next Friday. And so by next Friday, and the quiz is still similar as what we're doing, is just a question for options. But besides you gave me ABCD, I also need you to explain why. So that's the quiz. And the homework is basically asking you, based on the homework data set, you need to do the quality control. So first read the data, load the data, and then do quality control and normalization, and selecting highly variable genes, and the PCA, and clustering, and identify gene markers. Yes, and if you haven't done this, please fill in this survey, and it's important for our improvement. And thank you everyone for your attendance. Yeah, that's all. And for the rest of the time, it will be office hour if you have any questions.